Welcome to an introduction to genetics, our next topic. Genetics is a natural topic to cover next, cover after meiosis, because meiosis is the production of gametes, which are sex cells, the sperm and egg. And genetics is just the study of how those gametes combine to form new offspring and the traits that are found in the new offspring. There will be three different sections on notes for genetics. We will only be covering the first one today and before we actually get into class. The other two sections of notes will be handled in class at different times. Genetics was the modern genetics was first primarily outlined by one of my favorite scientists, Gregor Mendel. Um, we have known for a long time that there are traits and that traits are passed down. That can be seen when you look at families. You see families look similar. Children look like parents. But it was Gregor Mendel who first started putting the evidence together to support how we understand modern day genetics. He did not himself completely understand what he found out. It was not actually in his lifetime that what he found out was really made applicable or useful. However, it is his research which he published that did lead to our current understanding of genetics. Mendel's research was done on pea plants. He was a monk, and in his abbey he had a garden. And in the garden he grew pea plants. He kept records of the plants, and when he was going back through his records, he was able to start noticing some trends in the pea plants and the different traits and characteristics every year in the pea plants. And so he was able to do some mathematical calculations with these trends and determine um, some different mathematical realities that would happen if he did certain things to the plants. Now, it was sort of lucky that he was doing pea plants, that he was working with pea plants, because he didn't realize that pea plants have some traits which make them very good to use for genetic studies. He was just gardening with pea plants, and by just gardening naturally came upon and realized all these, or studied all these great traits. But it turns out, like I said, pea plants are basically the perfect specimen to use for his studies. And the reason why is that, first of all, a pea plant can self-pollinate. That means that it has both male and female gametes in the same plant. In fact, sometimes inside the same flower. So you can actually have the plant pollinate itself. Also, they grow very quickly and they're easy to grow, so you aren't losing a lot of new peas each year. Most of them are growing, and you can do your studies relatively quickly. You don't have to wait a whole ton of years to get a lot of good information. The thing that was really good about pea plants is that most of the characteristics of pea plants come in two very distinct forms. It would be like if humans only had two eye colors, if we only had blue eyes and brown eyes. Now we know that humans have many different eye colors, but if there were only blue eyes or brown eyes, it'd be much easier to study the genetics of human eye color. Well, in pea plants, there are a lot of traits which the pea plant only has really two distinct um, forms of. So they're either purple flowers or white flowers. Their stems are either long or short. Mendel actually ended up studying 7 different traits, and for all these traits, um, there are only two different sort of ways the trait can be expressed. So he was looking at the type of seed. It was either the form of the seed. It was either round or wrinkled. He was looking at the cotyledon. That's the very first leaf of the plant that's inside the seed. It was either yellow or green. He was looking at the flower color, which was white or purple. He was looking at the shape of the pea pod, which was either full or constricted. He was looking at the color of the pea pod, which was yellow or green. He was looking at the stems, and the stems would either have flowers on the axial stem, which is like the stem that's growing, or on the end of the stem. Those were the terminal flowers. And he was looking at the size of the pea plant. They were either tall or short. So for these pea plants, there weren't any middle colors. You didn't have a gray pea pod. You had a yellow and a green pea pod. So there are only two traits. Mendel's experiment was what's called a monohybrid cross, which means that he would only compare one characteristic at a time. So he would compare white flowers and purple flowers, or tall plants and short plants. He wouldn't mix them up. If you mixed up different plants, you would have a hybrid cross. Sorry, you would have a dihybrid cross or a trihybrid cross, and we'll work on those later. But right now, we're just going to look at what's called a monohybrid cross, which is where you look at one trait and you're crossing the, or you're looking at one gene and you're crossing the different traits for that gene. Here's just a look at the structure of the pea plant flower. The pistil, as you can see on the left, is the female part of the flower, and the stamen is the male part of the flower. So the pistil is the female part right here. The stamen is the male part. And Mendel was able to get the pollen from the stamen and put it on the pistil and germinate that flower. And so he was able to self-fertilize plants. And that'll be important in just a moment. When Mendel self-fertilized plants, he was looking to see if offspring looked the same as their parents. If Mendel was able to self-fertilize a plant and all the offspring looked exactly the same as their parents, it was called true breeding. That means that the true breeding means that all of the genes inside the plant code for the same thing. So if it's a tall plant and all the offspring are tall plants, that means that all the genes inside the tall plant are tall genes, or all the traits are tall traits. There's no small trait. If Mendel was self-fertilizing and he received some tall plants and some short plants, then he would know that the parent was not true bred. It didn't just have the tall gene. There are also short genes inside the parent. For Mendel's experiment, he started all of his parental plants with true breeding plants. He would find a true breeding plant, so he'd get a plant, he would self-fertilize it, and he'd look at all the offspring, and if all the offspring showed the same trait, he would know that he had a true breeding plants. So he would start with those so he knew what genes were inside that starting plant. He knew if it was all tall genes, all short genes, all purple genes, all white genes. So that's what you see right here in this picture at the very top left of the picture. In this example, we're looking at a trait of violet flowers and white flowers. In this case, he starts with a true breeding population of violet flowers and a true breeding population of white flowers. So that means that the violet flower, if it mates with itself, will always make violet flowers, and the white flower, if it mates with itself, will always make white flowers. This first generation we refer to as the parental generation, or the parents in the experiment. And so we signify as the P generation. when we talk or on the board or on a test, you might see a reference to a P generation. That's referring to the very first organisms that we start tracking in an experiment. In Mendel's experiments, his P generations, like we said, were true breeding. Now, the parents have some offspring, and we call those the F1 generation. F stands for filial, which just refers to sons or daughters, and this is the first filial generation. As you can see in the picture, the first filial generation only had violet flowers. So Mendel would get violet flowers and white flowers, and he would get the gametes from the violet flower and he'd share them with the white flower, germinate the white flower with them, and he'd get the gametes from the white flower and use them to germinate the violet flower. And no matter what, all of the offspring were violet. Now, after Mendel had an offspring that were all violet, he said, huh, what happened to the white flowers? Where did the white flowers go? So he got all the violet flowers in the F1 generation and he self-fertilized them. So he got those flowers, and he used their gametes to germinate themselves. And he received an F2 generation. This is the second filial generation. So we have the parental, which is the one that starts the experiment, the F1, which is where all of the, which is our first generation after the parents, and we have the F2, which is the second generation after the parents. And when Mendel self-fertilized the F1, all of a sudden he had purple or violet again and white again. But look at these colors underneath. In this case, he had 705 violet flowers in the F2 generation and 224 white flowers. He started with one violet and one white, and now he has 700 violet and 225 white, almost a 3-to-1 ratio of violet to white. So he has the violet and the white reappearing. However, they are not in equal numbers, and this made him start thinking about what's going on. In fact, this ratio, the 3-to-1 ratio, wasn't just for the one trait of violet and white flowers. Here's all the different things that he looked at: flower color, flower position, seed color, seed shape, all of them. And here are his raw numbers of how many offspring he got that showed each trait. Look at that, these big old numbers, lots of offspring he studied, and in all the cases, the ratio is almost exactly 3-to-1. 3 of one trait to 1 of another trait, all the way down. And so this made him start thinking, okay, so there's an actual pattern here, right? And so there's some control over the traits. Before Mendel, people would just say, okay, well, you have the essence of the mom and the essence of the dad, and those two essences mix and to make the children, who are just sort of like this weird essence of the mom and dad. They didn't know that there is a predictable way of determining genes of children or offspring. And Mendel started saying, wait a minute, these numbers can't be random. There has to be an actual mechanism going on here. There has to be something predictable going on here. In the F1 generation, that's right here, he saw some traits disappear. That would be the white flowers in this instance. But in the F2 generation, we saw those traits reappear. But they didn't reappear in the 1-to-1 ratio we started with. They reappeared in a 3-to-1 ratio, 3 of one trait to every 1 of the other trait. So how is it possible to explain this? Well, Mendel said, all right, organisms can carry both forms of a trait, right? But one might be masking over the other. So we know this organism is true breeding, so it only carries the violet color, the violet trait. This is true breeding, so it only carries the white trait. But this one right here, in order for it to make these children, must carry both the violet and the white trait. We just can't see the white trait, so we say the violet is dominant over the white trait. But once we self-pollinate, we can get right here where we see both the violet and the white. This is due to something called alleles. Alleles. A gene is something that we have in our body, right? So it's like the size of my nose. A gene is a trait, right? It's like the number of fingers I have, right? I have five. Almost everyone in the world has five genes, right? Or five fingers. That's a trait. It's our eye color. That's a trait. However, the trait doesn't just come in one condition. There are multiple versions of that trait, and each version is called an allele. So if the gene is eye color, blue eyes would be an allele. Green eyes would be a different allele. Brown eyes would be yet another allele. All of those are alleles of the eye color trait. Now, when creating sperm and egg, I have two alleles in my body. I have two alleles for every trait. I have the allele that my mom gave me and the allele that my dad gave me. So for eye color, I might have a green from my mom and a brown from my dad. So I have those two alleles. However, I can only give one of the alleles to my offspring. This is due to something called the principle of segregation. If you remember in meiosis, the very first thing that we separate is homologous chromosomes. The alleles are on separate chromosomes. One's from the chromosome I got from mom, one's from the chromosome I got from dad. And the first thing I do in meiosis is I separate those chromosomes from my mom from those chromosomes from my dad. That's the principle of segregation. And when I make my sperm or my egg, I'm only going to give one of those alleles to one sperm and the other allele to the other sperm. Or one of those alleles to one egg and the other allele to another egg. The sperm and egg will not contain both those alleles. Now the gametes can contain either a dominant allele, as we see right here, or a recessive allele, as we see right here. Now, when we're referring to dominant or recessive alleles, dominant alleles are always written as capital letters. Recessive alleles are always written as lowercase letters. So when we're looking at the pea plants, a tall pea plant is dominant. If the pea plant has a tall allele, it will always be a tall plant. So we say it's dominant. So we represent the tall pea plant with the letter T. Now the short pea plant is recessive. It will only be shown if there are short pea plant alleles inside the plant and there are no tall pea plant alleles. So we give the little t. So the capital letter is for the dominant, the recessive is for the small letter is for the recessive, and we choose T because that's the first letter of the dominant trait. So for color, if violet or purple are the dominant, we could use a capital V or a capital P. And if white is recessive, we could use a small v or a small p to represent that allele. Now, each of us carries two alleles. So what are the possible combinations that we can have? Well, we can have two dominant alleles, two recessive alleles, or a dominant and a recessive allele. And that's it. Those are the only possible combinations. Those combinations are called our genotype, or the genetic makeup of who we are. And as I showed, there are only three possible genotypes that you can have. You can have homozygous, where our alleles are the same, and if they're both dominant, we call it homozygous dominant. Or if they're both recessive, we call it homozygous recessive. Now, homo, if you remember, means same. And zygous refers to what's going to be given to the zygote, right? So it refers to the gametes. So this means our gametes, or what we had in our first cell, the alleles we had in our first cell are the same, okay? So homozygous, the alleles inside our first cell were the same. So we either had the dominant or the recessive. The other option is to have what's called heterozygous. Now in hetero, that means different, and zygous refers to our first cell, the zygote. So this means the allele inside our first cells were different. We had a dominant and a recessive. Now we don't say heterozygous dominant or heterozygous recessive because heterozygous always refers to having both of them. So the three combinations are homozygous dominant, the same dominant, homozygous recessive, the same recessive, or heterozygous, different. Something else to know is the phenotype. The genotype is the actual genes I have. The phenotype is what we see or how that gene is expressed. So if I'm homozygous dominant, capital T, that's my genotype. My phenotype would be that I am tall. If I'm homozygous recessive, little t, that is my genotype. My phenotype is that I'm short. If I'm heterozygous, I have the tall gene and the short gene. If the tall is dominant, that's why I gave it the capital, my phenotype is going to be tall because that's going to overpower the short gene. So let's go and look at this. Homozygous dominant, big T, what's the phenotype? Tall. Homozygous recessive, little t, what's the phenotype? Short. Heterozygous, big T, what's my phenotype? Tall, because tall is dominant. So let's look at this right here. Let's see if we can determine each pea plant's genotype for their flower color and write the genotype next to each flower. We know what the phenotypes are. Violet, white, violet, violet, and white. Those are the phenotypes. The question now is what's the genotypes? When you're doing this, you can stop the video now and try this yourself. I will go ahead and give the answers. But if you want to challenge yourself and see if you understand what's going on, it'd be good to pause the video now and see if you can figure out what the answers are. We're going to start with the P generation. Remember, these are true breeding, which means that they only have genes for the trait they show. So if I have violet flowers up here and they're true breeding, they're only going to have genes for the violet. So they'll be capital V, capital V. This is my white flower, and this is true breeding, which means that it will only have the genes for white, which will be little v, little v. And you might say, why are we using V for violet? How do we know that violet is dominant? Well, I'll get to that in just a moment. Violet can only give one gene to the next generation, and white can only give one allele to the next generation. So violet can give one allele, white can give one allele. But remember, it doesn't matter because the alleles they give are the same. Violet will always give the violet allele, white will always give the white allele. So when I get the violet allele and the white allele together, the plant is violet. So this plant right here has a violet allele, capital V, and a white allele, little v. Yet what color do we see? We see the violet, which means the violet is dominant. So this is capital V, little v. Now when I get this plant and I self-pollinate, it can give either a capital V or a little v to the next generation because it has both alleles. So we're going to start right here with the white flowers in the F2. Those white flowers, remember white is recessive, so they'll only be visible if the white allele is present, which means that it has to be little v in order to have white flowers. If you have a single big V, it's going to be violet. So what is the genotype of the violet flowers? It's a great question. We know that they're going to have at least one capital V. However, we don't know what the second allele is because if it's a two capital V's, it's going to be a violet flower. Or if it's capital V and a little v, it'll also be a white flower. So either of those are possible in this violet flower in the F2. So let's get to what's called Mendel's laws. Mendel didn't necessarily come up with these. He had ideas about them, but they were really determined later on once we understood better what was going on inside cells and what was happening with DNA. And once we actually found Mendel's research and we were able to apply his research to what we know about genetics. So the first law is the law of segregation, and this happens actually in meiosis 1. The law of segregation says that when an organism has a pair of alleles, only one allele will be given to each offspring. Or sorry, only one allele will be placed in a gamete. You do not place both alleles in the gamete. And remember, in meiosis 1, you get those homologous chromosomes and you separate them. So that's actually the law of segregation. So the law of independent assortment is a little bit more complex, and we won't really see it applied until we get to dihybrid crosses, which are about a week away. But the law of independent assortment says that when you have two or more pairs of alleles, right? So when you have more than one trait, those traits aren't dependent on each other when they are forming gametes. They don't really stick to each other. So what is this saying? What this says that if you get two particular traits for an organism, so if you get like the pea plants, the purple, so if you get like the purple and the white flower and the tall and the short plant size, the purple flower will not always be in the tall plant. It'll also be in the short plant. And the white flower will not always be in the tall plant. It'll be in the short plant also. So each of those traits is given to the next generation independent of the other trait. That's why we have such a mix of characteristics in humans, right? If I have brown hair from my mom, that doesn't mean I'm also going to have blue eyes from my mom. Right? My hair color and my eye color, those traits will be inherited separate from each other during the creation of gametes. One of the first people to really apply Mendel's experiments and his laws was a person by the name of Reginald Punnett. He is the maker of the infamous Punnett square, which we'll be using a lot coming up soon. Punnett worked on studying livestock, and he worked with a lot of farmers, right? And these farmers, they already knew that you could breed certain animals to get certain characteristics, but Punnett went about trying to actually scientifically determine what was happening so that there could be predictability in um, the mating and the heredity of livestock. So, what is a Punnett square? I'm sure you've seen this. It was probably in 7th or 8th grade. But a Punnett square allows you to predict the possible outcomes between two parents. So it says if I get two parents, what are the possible offspring that I could get? In this, what you do is you first all have to determine the genes of the parents. So in this case, we're going to look at the genes of a heterozygous parent, as you see right here with the capital T and the little t, and a homozygous recessive parent, the little t and the little t. Remember, it's recessive because they have the little t's. So each parent has two alleles, right? And what's going to happen is we're going to place one of the parents at the top of the chart and replace the other parent on the side of the chart. So the first parent is capital T, little t. So we always put the capital letter closest to this corner right here. So here's capital T, little t. If these are both capital letters, we could just write capital T, and that'd be okay. But if you have one capital and one little letter, you have to write the capital closest to this corner. The other parent is little t, so we write little t up top, little t on the bottom. And it doesn't matter where you put the little t's because both of those alleles are little t's. Now, we have the parent, we have the two alleles that make up the parent. Here's the parents and the two alleles that make up the parent. Now we're going to determine the possible genotypes and phenotypes of the offspring. I want you to keep in mind that if you have 4 children, that doesn't mean that these are what the 4 children are going to look like. This just simply tells us that each child has this percentage to be this result. So we drop the big T into this first box, big T. We push the little t over. Drop the little t, push the little t over. Drop the big T, push the little t over. Drop the little t, push the little t over. This right here is the possible genotypes and phenotypes of the children. So each child can be a big T, a little t, a big T, or a little t. Those are all the possibilities for each child. If you look, the child has a 50% chance to be big T, and a 50% chance to be little t. You can solve genetic problems using Punnett squares following a couple steps. First of all, you have to determine what the parents are. What are the parents' genes? Then you have to put the parents' alleles, right, based off their genes, into the parental part of the crosses. Then you have to fill up your Punnett square by dropping the top alleles and pushing the side alleles. Then you need to determine the ratios or percentages among the genotypes and phenotypes. So we're going to practice this. Here's an example that I'll work on the next page, but you can pause at this moment and see if you can work on this yourself. In pea plants, round peas are dominant and wrinkled peas are recessive. Using the steps above, cross a pea plant that's heterozygous for pea shape and homozygous dominant and a homozygous dominant pea plant. So what this is asking you is if you have a if you're talking about pea shape, right, which is wrinkled and round, what's the cross going to look like if you have a heterozygous pea plant? So that's big R, little r, because remember, round is dominant, so we're going to use that R, we're going to capitalize it. So big R, little r for wrinkled, that's the heterozygous pea shape. And homozygous dominant is going to be big R, big R. So if you have big R, little r, and big R, what are the parents' genotype and phenotype ratios or possibilities? Okay, so let's look at this. Step one, let's determine what the parents are. Well, here they're using capital T and little t and big T, big T. I don't like that. I like the big R, little r, because remember, we like to name them after the trait, but we'll go with the T's for right now. So here's the heterozygous P shape, big T, little t. Here's the homozygous dominant, the big T, big T. Step two, let's determine what the parents are. Well, or let's put the parents' alleles on the box. We have the big T, little t, and remember you always put the capital closest to this corner. And then we have the big T, big T. Step three, let's fill in the Punnett square to determine the genotypes among the offspring. So this, I'm going to drop the big T, push the big T. I'm left with big T, big T. Drop the little t, push the big T. Drop the big T, push the big T. Drop the little t, push the big T. I want you to notice also when you write these, you always put the dominant in front. So you never write this little t, big T. You always put the dominant trait in front of the recessive trait when in a heterozygous. All right, so let's determine our genotype and phenotype ratios. So how many, or what's the possibility that a child is going to be big T, big T? It is 50%. What is the possibility that the child is going to be big T, little t? It's 50%. So there's a 50% chance the child is going to be homozygous dominant, and a 50% chance the child is going to be heterozygous. Now, what is the percent chance that the child is going to be around? Well, if they have a dominant, they're going to be around. So this has a dominant, it's going to be around. This has a dominant, it's going to be around. This has a dominant, it's going to be around. That has a dominant, it's going to be around. That means we have a 100% chance that the offspring is going to be round. So genotype ratio, 50% homozygous dominant, 50% heterozygous. Phenotype ratio, 100% chance that they are all round. Here's some examples for y'all to practice with. So in people, freckles are dominant. Use a Punnett square to determine the allele combinations among a woman who is heterozygous for freckles and a man who is homozygous recessive for freckles. So freckles are dominant, so I'd say freckles is a capital F, and no freckles is recessive, so I'd do that as a small f. So one parent, if we have a woman who's heterozygous for freckles, she's going to be big F, little f, because that's homozygous. And if we have a man who's heterozygous recessive, that's going to be little f, little f. So we put the big F, little f up here, the little f right here, and fill out that box and see what you end with. In deer, albinism, being an albino, is a recessive disorder in which cells lack pigment causing the deer to be white. So being albino is recessive. So cross a homozygous dominant deer with an albino deer to determine which proportion of offspring will be albino. So first of all, let's determine what our alleles are. Our alleles are normal and albino. So I do have the dominant as maybe like a capital N, and the recessive, the albino, as a little n. So it says cross a homozygous dominant deer, big N, with an albino deer. Now, albino, in order to be recessive and see that trait, both traits have to be recessive, so that'd be little n, little n. So determine the parental cross, big N, little n, little n. Set that cross, fill it out, see what you're left with. The third practice. In sunflowers, fuzzy leaves are dominant over smooth leaves. Cross a sunflower true bred for fuzzy leaves with a sunflower that is heterozygous for fuzzy leaves. Okay. So remember when it says true bred, that means that it is homozygous. All right, it's only going to have the one trait. So if it's true bred for fuzzy leaves, it's only going to have the dominant trait. It's going to be homozygous dominant. I'm not going to help you with the parents on this one because I helped you with the last one. So go ahead and do your best to try to figure out what the parents are, set up the cross, and solve it. All right, let's talk about something called a test cross. We won't deal with this a whole ton, but it is something important to know and understand. We will have a little bit of practice on it. So if an individual's phenotype is recessive, we're going to know what their genotype is, because remember, you can only set recessive trait if that's the only trait present. If there's any dominant trait present, we're going to see the dominant trait. So example, in that deer, the albino deer, we're only going to see the albino if both of the alleles are albino alleles. If there's any dominant allele, we're going to see normal trait. Right? So that means that this albino deer is always going to be little a, if A is for the recessive allele. Now, let's look though at a normal deer. In a normal deer, can we tell what the genotype is? Well, this normal deer, it could be big N, right? It could be homozygous dominant, or it could also be heterozygous. So we can't actually tell what its alleles are, what its genotype is, just by looking at it. We have to do what's called a test cross. In a test cross, we know the organism has at least one of the dominant traits. That's why we can see it. We know this deer has at least one of the normal alleles, right? That's why we can see it. However, we don't know what the other allele is. It could be another allele or it could be an albino allele. So what we do is we get this deer, we get their alleles, and we cross them with a recessive allele. Why do we do that? Because remember, the recessive will always show the other trait if the other trait is present. So here's what that looks like. Here's an organism. We know their first allele. We don't know their second. We cross it with a albino. So we get the big A, little a, little a. Big A, big A, little a, little a. We know what this is. Now, if all of the offspring from these two organisms is going to show the dominant trait, we know that the second one is big A because all of the possible children are showing the dominant trait. Now, if this organism is heterozygous, then what's going to happen is half of the children will be showing the dominant trait and half will be showing the recessive trait because that recessive organism, that recessive allele, will show up again if this organism is heterozygous and has one of the recessive alleles. So this is called a test cross, where we get a organism that has the dominant trait, but we don't know its second trait, and we combine it with a recessive organism and we see what we come up with, and we compare that to what we should expect. All right, so I hope that this is a good start. We're going to cover more about this in class, but you should have enough notes at this point to complete your centers and study for a test. If you don't have that enough notes to do that now, you should probably go back and rewatch this and take more detailed notes, or reach out to me with questions, or research it through other resources online. The Amoeba Sisters or Bozeman Science are both really good resources for genetics and traits in most science topics. Um, so we will cover this in class. We will submit more information about class, but this ends the actual lecture period, and you should have really good notes at this point, and be ready to jump into this topic in class.